Professor?
Hi, good afternoon.
Good afternoon, Professor. Time ஆச்சுங்களா? 2 மணி ஆச்சுங்களா? ஸ்டார்ட் பண்ணிடலாங்களா?
Yeah, I'll start around 1:59.
ஓகே சார். Can you check the presentation, Professor? If you possible.
Yeah, maybe I can.
Check it, Professor. If you pass any troubles, we will face it, Professor.
I don't think that's issue because this we are doing every day.
Okay, Professor. Okay, nice, Professor. It's loading, Professor. Thank you, Professor.
Got it, right?
Okay, Professor. I got it, Professor. நான் program இருக்கமா நான் அப்புறமா.
So, how many students are attending this meeting?
Professor, average is 150 is attended, Professor. 240 minimum is registered, Professor. 250 is registered, Professor. Average is 150 is
Students.
Yes, Professor. Students came from Agri University, faculties, engineering backgrounds, science and technology, Professor. Both of national level, Professor. Some few participation from global international participation is there, Professor.
Okay.
Can I start, Professor?
Yeah, we'll start. Yeah.
Okay, Professor. Thank you, Professor. Welcome to Professor, scientist, students and research scholar. On behalf of organizer committee, RUSA Skill Development Certificate course, advances in data-driven statistical application and computational techniques. During the program February 12th to February 21st, 2021. The program is jointly associated with RUSA, Government of India and Government of Tamil Nadu. Today, the program is 8th day. The program is successfully completed. The supporting from University Authority, Honorable Vice Chancellor, most respected Registrar, and Professor Head, Department of Statistics, Bharathiar University. Today's special lecture, Professor Dr. M. Michael Gromika, Professor, Department of Technology, Bhubat and Jyoti Mehta, School of Bioscience, Indian Institute of Technology, Madras. Professor MSc from Madurai Kamaraj University, PhD from Bharathiar University. Sir research interest in discrimination of Merbourne proteins based on structure and functions, prediction on Merbournes of spanning of segmentation, alpha helical and beta boron membrane proteins, recognition of protein folds, protein of secondary and tertiary structures in globular proteins, conformation stability of proteins, and extreme stability of chromatic proteins, and so on, latest of applications. Professor's very high level achievement of associate editor, BMC Bioinformatics. The journal is the PubMed of level. The global level positions, SAR is associate editor. Professor has served in various committee for various academic, administrative and evaluation process. Sir is very busy schedule. Current year of GATE coordinator, IIT Madras. Sir is the coordinator. Sir is very busy schedule of time, lot of work. Now I request Professor Dr. M. Michael Groman, IIT Madras. Welcome you, Professor.
Okay, shall I start?
Yes, Professor.
Yeah,
Professor, any update I missing, Professor? Can you for your latest and current update task is himself, Professor.
okay, maybe I'm not a gate coordinator because I have some gate duty, but
Okay, Professor.
yeah, good afternoon, and thank you for inviting me to give a talk in the skill development program. And am I audible?
Yes, Professor.
Okay. Yeah, I'm sure you might have learned several aspects of the skill development and the computational algorithms in various fields of science. And my focus is on the biological aspects. So I'll discuss mainly on the what are the how the data-driven approach works for understanding protein structure and function. Okay. So considering the students, so may I go this little bit slow, and I mainly my talks are kind of a discussion. So I ask questions. So if it is good, if you answer the questions quickly, right? And also I start with some basic fundamentals, assuming that most of you are not familiar with the topic because it is completely biological aspects, how we use these computer algorithms and computation techniques as well as the computer resources to understand protein structure and function. Okay. As Professor Gandhiya mentioned, actually I belong to the Department of Biotechnology, IIT Madras. It is founded in 2004, and currently we have 29 faculty members, and a couple of them are joining now. And we have more than 500 students, and we offer dual degree BA, BS, MS, and B.Tech, M.Tech programs, and MS by research and clinical engineering, M.Tech, as well as the Ph.D. programs. We mainly focus on 4 aspects: bioprocess engineering, chemical biology, computational biology, and molecular biotechnology. Chemical biology we recently renamed as biomolecular sciences. And computational biology, we have strong background and several faculty members and working on different aspects, such as structural bioinformatics, computer economics, molecular biophysics, neuroscience, and structure-based drug design. Right? We have several PhD positions available, especially for those cleared GATE or CSAR. Right? And we encourage the applications are available online all the time. So encourage the students, if you are interested, to apply, and we have positions right available. And also we have several projects available, and if you are interested, you may contact or check our websites, and then you can contact the faculty members who are having positions in that. Okay, in computational biology, so I work on the protein bioinformatics lab. So this is my lab members, and we have a beautiful campus. If anybody wants to visit, just let us know. We'll arrange for that. And we have good computational facilities, and we have very natural sceneries and so on. So coming to the research, okay, I am working almost different aspects of proteins to understand the structure, folding, function, stability, as well as interactions. This is, I see in this slide, I summarize most of my research topics. It is too much research oriented, so I don't want to go to details for all the aspects, but just I want to touch a couple of things. One is we are working on the protein folding and stability. So we developed the concept of LRO, that is called long range order, which relates the contacts of amino acid residues in protein structures, how the amino acid residues in protein structures, right? I will give more details maybe in the coming slides, right? Interact to each other, whether they are making short range contacts or medium range contacts, or they are far away in the sequence and they have the contacts in 3D structures, right? So based on that, we propose this concept. And if we have high long range order, then it will slow down the folding process because it has to make large number of long range contacts. If it is less, then it will the proteins fold fast, right? Because it has to make very less number of long range contacts. On the aspects of stability, we developed database called Protham, the thermodynamic database for proteins and mutants, right? So we got more than 1,000 citations for that. So it's a crucial database and it's a very important database. It's the only unique resource in the literature, right, to get the reliable data on protein stability. So based on that, maybe hundreds of methods they developed in the literature, and we also made several methods to predict the stability upon mutation using protein 3D structures called CubSat and protein sequence, that's IP3, and the stride to identify the residues which are important for the stability. Fine. Then you go to the membrane proteins. So there are two types of membrane proteins, alpha helical and parallel membrane proteins, and we try to understand the specific features of these type of proteins. And if we have a genome sequence, is it possible to identify these type of proteins? If you identify these type of proteins, is it possible to identify the residues which are spanning inside the membrane, that is in these regions, right? And how far is related to the functions, right? Whether we can find any specific motif to understand the function and so on, right? And then we did the protein aggregation. So because this also involved various diseases, especially neurodegenerative diseases. So we want to understand the mechanism, whether we are able to identify the aggregating peptides or the proteins, right? And how this is causing influence in different diseases. Okay, then we did the protein interactions, protein interactions, protein nucleic acid interactions, protein carbohydrate interactions, and protein ligand interactions, that is structure-based drug design. So we developed databases to collect the experimental data on binding affinity and the factors which influence affinity, and we are able to predict the affinity and whether we can do the large-scale network analysis. We can do that, whether we can distinguish the high and low affinity complexes and whether this is related to specific functions and the specific disease and so on. Fine. Then recently we started the analysis and big data analysis, like we did the transcriptome data analysis for the Alzheimer's and Parkinson's diseases. Currently we are working on the single cell analysis with the ALS and then Huntington's disease, and that's also we are focusing to understand the variants and the pathways networks and to see how can we relate with respect to this experimental data. Then we are focusing various applications with respect to different diseases, like the protein-protein interactions in malaria and protein aggregation in Parkinson's disease and EGFR mutations in cancer and the different in dengue, chikungunya, and so on. So one of the major aspects I can tell you that in BCL2, we developed a computational algorithm based on data-driven approach to identify the potential inhibitors, right? And then we did the experiments, various biochemical assays to validate the identified inhibitor, the compounds, and did the animal studies, right, to see whether first we gave the injector the tumors. So it was so increasing, but when you give our compounds, it decreased, right, and in a specific period of time. So there also we did with animal studies, and we found that our compound was fine. So that means whatever we do the computational aspects in bioinformatics and computational biology, right, we can easily test the experiments, right, and we can validate our data with the experiments. Fine. So I think I'll start the lecture. So when you talk about the proteins, right? So what do you think about proteins? There are various biological macromolecules, proteins, nucleic acid, and carbohydrates. Right? So what do you think about proteins? Your lunchtime is good to sleep, so just give me some answers so that you can be active. Once nobody heard about the word proteins. Right? Okay, yeah. So if you not familiar with the proteins,
Yes, sir.
okay, help to build muscles. Okay, any other answers? Good. Yeah. So proteins perform diverse functions in living organisms, right? So you can say the enzymes which catalyze the reactions, right? So there most of all the enzymes are proteins. There are several residues which are in the active site, right? So they will enhance the speed of the reaction, right? And you can see the transport proteins, the proteins which transport oxygen, right, from the lungs to the other different parts of the body, right? That is important, right, for the transport proteins. If there is a change of any amino acids in these proteins, like the hemoglobin, right, and there is a disease called sickle cell anemia, that is for the breathing problem. If you mutate a particular residue, the glutamic acid to valine, right? So it is a transfer proteins, and we have the antibodies, because currently people are in the COVID-19, so people are talking about antibodies, right? So we have several antibodies, immunoglobulins in our body, which can fight against this disease, invade of bacteria and virus, right? And we have these several proteins, which are the structural proteins that give the strength, that mainly we can see the keratin or the myosin. And we have the sugar metabolism, that's insulin. So we have this metabolism, and we have thrombin, we have the blood clotting protein, right? So there are various functions. Proteins perform diverse function living organisms. Also in diets, you can see the albumin, you can see this in the casein, in the milk, so you can the egg white. So there are several proteins which are in the diet, right? So among the three biological macromolecules, nucleic acids carries genetic information. Carbohydrates is a staple of diet, may they call the glucose and fructose, as well as the cellulose has the strength or the protective muscles, and the proteins also perform diverse functions. Okay, so when we talk about the proteins, there's a building block of proteins. So proteins consider 20 naturally occurring amino acids, right? These amino acids are the building blocks of proteins, right? So why it is the amino acids? Because if you see here, so we have one NH2 group, that's amine group, and there's a carboxyl group, that is COOH group. So that is called amino acids, right? So there are 20 naturally occurring amino acid residues, right, in a protein, right? So these 20 amino acid residues, which are majorly classified in two groups. One group is called the hydrophobic residues. These are the residues, right, which hates water. That is what literally, if you talk about that, is phobic means it is hating hydro means water, right? So another group is hydrophilic, that is water loving amino acids. That means the residues, they like to interact with water molecules. So if you see the hydrophobic residues, this is mainly grouped into three different subgroups. One is the aliphatic, one is aromatic, and sulfur containing. Right? If you see the building blocks, right, if you see it here, so this is the common for all amino acids, NH2, C alpha, COH, and H. This is R. This R is different. The R there is 20 different types for the 20 different amino acids. Because here NH2, C alpha, COH is commonly occurring in all the amino acids, right? And here this R group changes. In glycine, this is the simplest amino acid. Here the R group is H, right? And all others, the next one is the alanine. That the side group is CH3. So then you can see all these aliphatic amino acids contain aliphatic groups. So there's alanine, leucine, isoleucine, valine, right? So these amino acid residues represented with the names are 3-letter codes or single-letter codes, because these are very commonly used by the literature. So they have the 3-letter codes and the single-letter code for 20 amino acids. Like how we have 26 the alphabets in English, right? The aromatic residues, if you see, so these residues contain the ring. Okay, see phenylalanine, tyrosine, tryptophan, they have ring structures. That is the reason why it is called aromatic residues. And sulfur-containing residues, this contain the atom sulfur. One sulfur is here, this methionine. This sulfur is here, that is cysteine, right? This is sulfur residues. These are all the hydrophobic residues. Fine. So on the other hand, we have the hydrophilic residues. These residues tend to form hydrogen bonds, electrostatic interactions, right, van der Waals interactions, all these interactions. So these are the classified 3 different groups. One is negative charge. Right, we have the COOH group here. This is aspartic acid and glutamic acid. And the positive charge, this is amine group. Here there is an NH here, NH2 here, NH2 here. And the polar residues, so you have the CONH2. Here this is the glutamine, and this is alcohol group OH. This is the 3,1,N CHOH, and here is the proline. And these are the polar residues. So the residues are broadly classified into two groups. What are the two groups? You may unmute your mic and then tell, or you can write your answers in the chat box. Both are fine. Right? What are the two groups of amino acid residues? Okay, so let's move forward. And these amino acid residues, right? Okay, you can see this is amino acid 1 and this amino acid 2. Right? Here this side group is R1. Here this is R2. Right? When they join together, so with the elimination of water molecule, this COH is this OH and this NH to the H, they eliminate water molecule. Then finally there's form a bond that is called peptide bond. Right? So here now this is the main chain, right? And this is a side chain. Side chain is different, but likewise the different amino acids, right, join together by the elimination of water molecules and the formation of this peptide bond. They form a protein chain, right? So if we talk about these proteins are of different categories, right? So one is the globular proteins, and the membrane proteins, or the fibrous proteins, and these proteins they have different functions, right? So how the proteins are formed is like as I discussed earlier. So this combination of these specific amino acid residues, right? They eliminate water molecule and form a specific peptide bond, and the different combination of these residues they can form a structural functional protein, right? So I have a question: is it possible to make any combination of these amino acid residues to form a functional protein, or only specific combinations are possible? Right? Okay. So like the English alphabet, you got 26 letters, but you can make any combination of 26 letters, but that words or the sentence you cannot find in a dictionary. Right? You can put 5 A's and 5 E's, right? But you can't form any meaningful words, right? Likewise in the proteins, so the combination of these 20 amino acid residues, right, there is specific combination of residues, right, to form a functional protein. You can make any combination, but that is not a functional protein. That is called the polypeptides. But to have a functional protein, nature selects a specific combination of amino acid residues for a functional protein. Fine. These proteins are different types, right? One is the globular proteins, right? One is fibrous proteins and membrane proteins. Globular proteins perform various functions. Okay, I'll explain some of the functions in the following slides. Fibrous proteins, as one of you mentioned about these muscles and all. So these are the proteins which are involved in the structural proteins. Membrane proteins, the proteins which are embedded in membranes. So there is a membrane between the cytoplasm and periplasm, and the periplasm outer space. Right? The proteins which are embedded in this membrane are called membrane proteins. Right? I guess a little bit introduction about the proteins. After I'll go to these computational approaches. Fine. So these proteins have the helical conformation. This is called helical proteins, and this one the beta strand conformation. So this is called beta viral proteins. Fine. So as I discussed earlier, so we have the various functions. Like the enzymes, these are specialized proteins with catalytic activity, right? That is increase the rate of a chemical reaction. And small portion is enzyme, right? That's the 3 to 4 amino acids directly involved in the catalysis. And this site that's called active site. And the transport proteins, as I discussed earlier, hemoglobin, right? Present red blood cells efficiently carries oxygen from lungs to tissues of the body. And defense proteins called antibodies, regulatory proteins, structural proteins, channel proteins, receptors, and so on. There are various functions. And these functions, which are mainly dictated by the structure. How the structure looks, likewise you can see that if you tell the friends of some person, you can tell the character of the particular person. Like that, if you know the structure of a particular protein, then you can easily understand the function. So most of the functions are dictated by the structure of a protein. This is one of the famous tumor suppressor protein, the P53, right? It interacts with the DNA, and here this portion which interacts with the DNA, that is very important. And if you mutate these residues, that lost the activity, right? So that lost the function, that causes the various types of cancers. Okay, I'll tell you the different types of structures. The protein structures are mainly classified into 4 different groups, right? Only the primary structure. Primary structure is the combination of the different amino acid residues, right? You can see the different amino acid residues I showed in the first slides, right? Just we can give the combination of these residues, right, to form a functional protein. And the second level, right? So you can see these residues which have some periodic arrangement, right? Here is a kind of the spiral shape, and here down you can see it's a kind of the ladder shape. The spiral shape structure you can call this alpha helical structure because it is first formed in the proteins, and the first Greek word is alpha, so it is alpha helix, right? And the second thing is they form this barrel structures, strands, right? Like the ladder type of structure, that is called beta strands. The tertiary level tells you the exact location of each atom, where each atom in a protein is located in the 3D structure. Right? There are various experimental techniques like X-ray crystallography, NMR spectroscopy, like the current cryo-EM. Right? These methods have been widely used to determine the structures of these 3D structures of the protein. And the fourth level, I can say the quaternary structure. This is the assembly of subunits. The various 3D structures forms the quaternary structure. With the 2 forms, this is dimer, 3 means trimers, right? 4 means tetramer, and so on. Right? And depend upon whether these two are same, they are homo, or if the 2 subunits are different, that's called hetero. So you have the homodimers, heterodimers. Essentially, you can say that homo oligomer, hetero oligomer, so on. Right? So we go with the primary structure. Right? Here you can see the specific combination of these amino acid residues, right? So form the structural functional protein. So this is a sequence for the human hemoglobin subunit beta, right? So here you can see the glutamic acid here, right? E. If you mutate to valine, right, this causes the sickle cell anemia. That means the specific combination of these residues is required for its function, right? If you mutate any residues, or if there is any changes in this sequence, right, that may form various diseases, right, in human. Right. So now to derive data, right? So we need to have database, right? Because to have the data-driven approach to understand the structure or function, right, we need data, right? So these sequences, right, of the proteins, right, they are stored in various databases. Right. Here the main databases are called the PIR, that is Protein Information Resource. This is the one of the first developed databases. Margaret Dayhoff from Georgetown University. She collected the data from the literature, and she published various books, atlas of protein sequences, right? And later on, they changed to the database called PIR, the Protein Information Resource. In the meantime, Swiss Institute of Bioinformatics in Geneva, they also collected the data and developed this database called SwissProt. After several years, right, they joined together and they made a consortium so to develop this UniProt, that is a universal protein knowledge base, that is the unique resource for protein sequences as well as the functions, right, to give all the information regarding any particular protein. So currently it is a unique resource, the UniProt, the universal protein knowledge base, which contains data for any protein, right? It has about 7 million sequences, right? So this is the UniProt database, right? So it is provide high level of annotation, such as description of the function of the protein, domain structure, and variants, and so on. A minimal level of redundancy and high level of integration of databases. Right? There are 2 different types of data. One is manually curated, and another one is computationally annotated. Totally, this is April 2020. At the moment, we have 207 million sequences. It's available as of now. So human manually curated data is almost the same, because now currently actually less number of people are involved in that. So computationally translated sequences, that's very high. We have 207 million sequences currently available in UniProt. If you look into this UniProt database, right, the average length of the proteins is about 200 to 400 residues, and the 315 residues is the average length of all the sequences deposited in the UniProt database. And if you see the preference of residues, some residues are highly preferred. For example, these aliphatic residues, there is a gray color, that is alanine, valine, and leucine, and so on, and followed by this red color, that is the charge residues, and the green one, right, the polar residues, and so on. Right? Then some of them are highly occurring because to the form the hydrophobic core and for this make the protein stable, right? And some of the rarely occurring amino acids. Okay, so I gave a sufficient introduction. Maybe then I'll go with this computational point of view. Okay, if you have the sequence, I gave one sequence here. You can see the considered as 26 alphabets in English, right? What can we do with this? If I give this to you, right? So what are various aspects you can think of? I need your response. Can you tell me what are the information you can derive, you can get if I give a sequence like this? Any idea? You may unmute your mic or you can give a response in the chat box. Now, if I give this sequence, okay, what are things you can calculate?
Professor, some people post the chat, Professor.
Yeah, you can just count the number. You can say this contains this many letters. Second is you can count how many A's here, how many D's here, how many C's here, right? Then you can see which letter comes quite often. Right, all these things you can count the numbers. How many letters in the sequence? That means sequence length. Okay, so first one is you can see the for example, if I give this one, how many letters here? Just you can calculate and count this. Right. So then if I give this is the sequence. For example, if you see this is the sequence, for example, how many A occurs in this sequence? Because these all will have the biological meaning. So how many A's in this sequence? This is very short prepared. I give TH, IS, IS, A, PE, PTA, DE. How many A's here? Are you able to see my screen? Yes or no? I need some response. Yes. How many A's in this sequence? We take from T to D. T, H, I, S, I, S, A, P, P, T, I, D. How many A's in this sequence? One. Good. Very good. How many C? Very good. Right. So this is actually I made this for the simplicity because so that you can easily calculate. Right. Good. There are no C's. Right. Actually, down if you see that there is actual sequence. This actually protein sequence. So if you see this sequence, you can calculate how many A's, how many D's, how many C's. Right. So if you see the down, I gave you A 15 times A occurs, C 2 times. There is no B in the amino acid. D 7 times, E 8 times, and so on. Because this combination will tell you specific functions of a protein. For example, if you talk about proteins are enriched with the hydrophobic residues in the membrane. So if you see there is more number of leucine or valine or alanine, you can say this could be probably the transmembrane helical protein. If you take a DNA binding protein, this is these proteins mainly enriched with the positive charge residues because the nucleic acid is negative because of phosphate group. So these positive charge residues like to have interactions. That means if it is predominant with the lysine and arginine, lysine is K, origin is R, then we can see that this could be a DNA binding domains, DNA binding proteins, right? So these numbers is not simple joke. These numbers give you some sort of information to a particular protein. Okay, so now this is the first thing I can calculate occurrence. This is called amino acid occurrence. Occurrence will tell you how many times each amino acid residue occurs in a particular sequence, right? But in this case, one disadvantage is one protein has 100 residues and another one has 1000 residues, right? In this case, naturally 1000 residues protein which got more number of these residues. So in order to compare these two, the numbers won't help because that depends upon the size, right? Then how to do it? How to eliminate the influence of the size? We can do simple math, right? Because one protein is 100 residues, one protein is 1000 residues. The numbers will be different, the occurrence will be different, right? How to eliminate this bias? Right, what to do to eliminate this bias? For example, I ask a question. If there is 100 residues and 10 are alanine, right? What's the percentage of alanine in this particular protein? What's the percentage? Okay, so this will give you the occurrence of each residue in a protein, right? So in order to consider the length of these proteins, right, we just normalize with respect to the length. In this case, you can get the preference of these amino acid residues in a particular protein, right? For example, if you have the in this case, we have 15 alanine. Number of residues is 147, right? So in this case, you can calculate the composition of alanine. That is 15 into divided 147 and multiply 100 to get the percentage. So it is 10.2 percentage, right? Fine. So then, what is the percentage of the cysteine? You can take this is 2 divided by 147 multiplied by 100. So that will give you this percentage of this composition of cysteine. So for each amino acid residue, right, you can get the composition, right, in a particular protein, right? If all the how many amino acid residues, naturally occurring amino acid residues? How many naturally occurring amino acid residues in a protein? $20, right? Fine. See, if these 20 residues are equally distributed, what is the percentage of each amino acid residue in a protein? If 20 residues are equally distributed, what is the percentage of each amino acid residue in a protein? 20 residues. 20 residues are correct. If the 20 residues are equally distributed in a sequence, right? For example, take 100 residues in a protein and 20 residues, all are equally distributed. Then what is the percentage of each residue? This is simple math. Are you able to follow me? Because 20 amino acid residues, right? 100 residues in a protein. If all are equally distributed, how many times each residue occurs? 5, right? Fine. Yeah. So if they equally distributed, right? So now you can see the 5% is the average number. But if you calculate it, okay, so calculate it, right? So here these are the 20 residues, right? So here there are 79 residues, right? So when you calculate the composition, right? Can you see the preference of any letters here? Can you tell which now letters are overrepresented, which letters are underrepresented? A is overrepresented. A you can see 21% here. Very good. Is overrepresented, and you can see some residues underrepresented. Which residues are underrepresented? V valine, yeah, valine is less, correct, and Q is less. Yeah, this is underrepresented. So this is a specific behavior of this particular protein. Right? So based on these residues, the occurrence of the residues, we can understand the functions. We can understand. I'll tell you in a few minutes how this composition is very important, right, for to understand the structure, understand the function. Okay. Now, if I give a sequence, can you write a program to calculate these numbers, this composition? What are the input data you need? So I ask you to write a computer code, a program, any language, right, to get this composition. How to do this? Right, first you read your sequence, right? This is your sequence, right? Okay, sequence is here. So you need the sequence, right? There are 20 different alphabets, so you can just put an array that 20 different alphabets and initialize all these 20 values, right? And take the sequence and you compare with the 20 alphabets, and if it matches with the matches, then you add in that particular array. If not, repeat. So go up to the end of the sequence, and now you can see this NO of J. They will give you the numbers, right? So we'll give this get these numbers, right? When you normalize with N, normalize total number of residues, right? Then you will get this composition, right? So this is how you'll write a code. So we can write a simple code to calculate the number as well as the percentage of each residues in a particular protein. Fine. Okay, now we have I discussed about the PIR, right? So if you want to have it, you can take any sequence, right? And you calculate by yourself, write a code and calculate, and go to this PIR, and there is a tool called search analysis. Go here, and here there is a option called composition molecular weight. Click here the composition. We end up with another window. So here you write your sequence, any sequence you give here, your sequence, and click on submit, right? Submit if you click, right? Then you get these numbers. The same I showed you earlier, right? So these are the 20 different amino acid residues, and here these are frequency. Here you can calculate. Okay, next up, I discussed two aspects. One is the occurrence, and the second is the composition. And the third aspect, that is called molecular weight. Right? Molecular weight you can calculate using this formula. N of I multiplied by W of I minus 18 into N minus 1. So you know why you put minus 18 into N minus 1? this Wi for each amino acid, for example, 5 times ALA occurs, multiply with the 5 times of ALA, weight of the alanine. 10 times D occurs, multiply 10 times the weight of this D, and you can add up to get this number. And finally, subtract it to the 18 into n minus 1. Why do you want to subtract this one? Any clue? How we form a dipeptide? When the two amino acids join together, what will happen if the two amino acids join together? Water molecule is released, right? So what is the molecular weight of water molecule? (18), right? This is what this 18 comes here. Why it is n minus 1? Because when you combine two amino acid, how many water molecule will be released? If you combine two amino acids, how many water molecules will be released? 1. If you have 2, 1. If you have 3 amino acids, how many water molecules will be released? 2, right? So there's n amino acids, there will be n minus 1 water molecule will be released. So that is why I put 18 into n minus 1. Fine? Okay. So now I have the numbers. Okay, this is the 20 amino acids. We have this molecular weight, right? Because how we get this molecular weight? Because you have the carbon, hydrogen, nitrogen, the values. You can take it and then you can, this is the molecular weight, right? And the water molecule, we put 18, right? So for any sequence, I gave you sequence earlier. So that sequence, right, you can calculate your molecular weight, right, for this particular sequence minus, you remove the water molecule, then you will get the molecular weight of that particular protein, right? Fine. So that is one aspect. So you can easily calculate the molecular weight. So another aspect is you can calculate the average values for any properties. So the proteins and peptides, they have many various properties. They have the polarity, they have the charge, they have hydrophobicity, and they have the molecular weight, they have shape, size. So there are various properties. So you can calculate total properties, or you can take the average properties using this equation. N of I multiplied by any of these properties. Right? So for example, I give these numbers. Okay. This ACGMY1, FILVM2, W2, this is minus 2 minus 1. Right? Can you calculate the hydrophobicity of this peptide 1? Take the question number 1 here. Right? What is the total hydrophobicity? The value should take from here. And can you tell the number for the 1 and for the 2 for 2? This will distinguish different proteins or the different peptides. So several properties which can distinguish one peptide from other. Right? So this is one example I mentioned here. Right? What is the total hydrophobicity for this peptide 1? What is the total hydrophobicity for peptide 2? Can you get the numbers quickly? What is the total value for what is the value for A? One, right? What is the value for M? Now, what is the value of E? M is 1, A is 1. The total value is, what is the total value? Add up all the numbers and get the numbers. 1 plus 1 minus 2 minus 1 plus 2 minus 1 up to 0 is 1 minus 2. So what is the value for the total for this number 1 peptide? Substitute the values and give me the number. Can you give me the numbers? Because that should be useful to the participants rather than if I go for 1.5 hours, but if you don't understand anything, that is totally useless. Tell me a number quickly. What is the value for the peptide 1? Anyone calculated the number? Good. Yeah, minus 4 and 13. So you can see the difference. So first one is a minus 4. Minus 4 tells you it is enriched with the charge and polar residues. And 13 will tell you this is enriched with the hydrophobic residues. If you see these numbers, so these are all hydrophobic residues. That is why I put 1 and 2. And the minus 2 is mainly the charge residues, and the minus 1 is polar residues. With minus 4, that will tell you this peptide is mainly contains the polar residues and the charge residues, and have the high potential to interact with the DNA. Right? And the second one, you can see highly hydrophobic because the number 6 is 13. That means highly enriched to the hydrophobic residues. So that could form a potential transmembrane segment. That means the segment which traverses the membrane in the case of membrane proteins. Right? So we can tell these numbers some of the characteristics of a particular peptide, or we can tell characteristics of a particular protein. So that is the reason why we give these numbers. Right? This is not just random numbers. Fine. So then we developed a tool, right? The sequence to feature. So here, this is we published in Bioinformatics 2019, right? So if you give the sequence, right, give any sequence here, or you have the option to upload the file. Okay, this is the website. Okay, this is my website here, and you can get this information. Right? If you give the amino acid properties and submit here. Okay, these are the various properties, right? I mentioned this website, and you get this actual value. And here we can normalize between 0 to 1, right? So you know how to normalize between 0 to 1. So if you normalize, this is average value. From these numbers, you can tell that, okay, which one this sequence is enriched with what, right? This 0.4, that is less than that. This 5.58, that means it has more medium range non-bonded energy. This is 7. The pK, that is this acidic property because it is 0.7. If you take the hydrophobicity, should be less. So you see it is 0.4. That means it contains less number of the hydrophobic residues. Right? These are the information you can get from this particular sequence. Right? You can get the information. Okay. Next question is why the composition is very important. Why we are talking about the composition? Why the composition is very important? Right? I give you 3 sequences, right? And some letters I highlight in red. Can you tell what is the importance of the residues which are highlighted in red? So I got 3 sequences, 1 PRC, 5 acro, and 2 4. Can you look at the sequences and tell me why some residues are highlighted? You will take the last one, 2 POR. There is question number, there is a sequence number 2, right? Here if you see acidic. Any other answers? Among the 3 sequences, sequence 1, sequence 2, and sequence 3. Any other answers? Just you look at the highlighted with red and just give the answer. That's all. Last one is very simple. You can see all the yes I highlighted. And the middle one, which letters are highlighted? R and K, right? These are basic residues. This is it has NH2 groups, right? So these are the positive charge residues, right? And this is why I highlighted R and K. The first one, can you see the highlighted residues? They have any specific characters? Okay, so these are all hydrophobic residues. Okay, so all these residues, alanine, isoleucine, phenylalanine, all these hydrophobic residues. So that is the reason I highlighted here. If you put these three types of sequences, they have some specific special meaning. So this is the membrane protein, alpha helical membrane protein that's enriched with the hydrophobic residues in the membrane. And the second one is a DNA binding protein. They are enriched with positive charge residues to interact with the DNA. And the third one is a beta bar membrane protein, which is enriched with serine to form the hydrogen bonds. Okay, so they have some physical meaning and they have some functional context. That is the reason I put these three sequences with different colors. Okay, so if you see the two groups, for example, I put transmembrane helical and the transmembrane beta strand. The two groups, one is a globular, one is a YMP. Right? So we calculate the composition. Right? How to calculate the composition? What is the formula to calculate the composition? If you give a particular sequence, how to calculate the composition? So you get the occurrence for each amino acid, normalized with the total number of residues, you get the composition, right? So here we get a set of proteins, right? So we get the occurrence of each residue, right? All the 20 residues here. I given the 3-letter code and normally 3-letter code and normalized with the total number of residues, right? And this is the your composition. So if you compare, I give two values. One is for the globular, one is for the YMP. Can you see any significant difference for any of the residues? Some cases the data are almost similar, right? For example, if you see 8 point leucine, 8.48, 8.78, 8.47, 8.95. Some cases is dominant in globular protein. Some residues are dominant in YMP. Which are dominant in the globular proteins? Write the codes which residues are dominant in globular proteins. Are you able to see my slide? Correct. Yeah. This is cis and the glow. This is very good. So these are dominant globular proteins. On the other hand, if you see some residues like if you see here the serine, right, or asparagine, okay, these residues dominant YMBs, right? So you can see the difference. So I show the figure. So here easily you can understand the concept, right? So down one is this mainly dominant in the case of the YMP, and the top one which is mainly dominant in the case of the globular protein. Some cases you see the serine, if you see there is very much difference between the globular and the YMP. So it's very high difference, right? So now we can use this information. Some of them is very less. Okay, we have this one and this one, does not make difference. So it doesn't matter if you take this information content, right, for your analysis or not. Those are high difference, like here the serine and this glutamic acid and this histidine. Okay, these things, right? It has high influence to discrimination. Now the question is, if I give two sets of proteins, group A and group B, based on this composition, are you able to discriminate? Can you tell this new protein belongs to group A or belongs to group B? Right? So I tell the algorithm. Okay, so first we have the set of proteins belong to group A. We get the composition. That's what I showed here. Okay, here this is the group A, if you say that. And B, take the set of proteins and you get the composition, like the composition of B. Then take a new protein. We don't have the protein X. The question is, now we need to say whether it belongs to A or B. Right? So get the composition. Then you compare this composition with respect to the composition of A and with respect to the composition of B. Right? With this average value, so you get the total, this is equal to 1. So you can see the composition of A minus this composition of this X. X we don't know. That is, we put the absolute value. Why you put the absolute? What will happen if you don't put the absolute value? So what will happen if you don't use absolute? If you take the number as it is? Any answer? Okay, so differentially maybe negative. Any other answers? Because usually any other answer for this? Why you need absolute? Yeah, if you don't take the absolute, that will nullify. In some case, A is high, composition A is high. In some case, composition H is higher. Then when we get the positives and negatives, finally they will cancel out. Right? So, but our aim is to get the difference. Right? For example, if a composition of A is 5 and the X it is 4, the difference is 1. And for the next one, if this is 4 and another one is 5, the difference is 1. But if you take the signs as it is, 1 minus 1 equal to 0, that will be 0. Actual difference is 2, 1 plus 1 equal to 2. Right? For that reason, we take the absolute value. So now we do it for all the 20 residues, exact the difference, take the absolute difference and add up, and that is the average, that is a sigma. That's a deviation from A. Deviation of X, right, with respect to A. Right? The same way you can do the difference of this X with respect to B, right? Then we can see the sigma of B, right? Then you compare the sigma of A and sigma of B, which one is lower? For example, B is lower, then there is belong to group B, and if A is lower, it belongs to group A. Right? This is simple concept. Right? We can say the composition, easily you can calculate the composition, get the current normalized with the chain length. Right, for two groups, for the new one, get the composition, compare with the group A, compare the group B, and get all the sum of all the differences, and which one is the lowest difference, right, then it belongs to that group. This is one aspect. Second aspect, you can take the correlation. If the correlation is high, you can take it as belongs to a particular group. There are various ways to do that, right? That's kind of research problem, we can do that. Okay, now I give one example, right? So here, this is one protein, this is called the globular protein. This is the YMP. So take the composition. This is the number of occurrence. This is a composition. This is the difference with the globular protein. It's a difference with the YMP. So then compare these two. This is the lowest one. So this is grouped as globular protein. Right side, if you see, this is the difference with the globular protein. This is the difference with OMP, and if you see the difference, this is the lowest one, so it belongs to outer membrane protein, right? So we can do it with various set of data, and you can calculate the sensitivity, specificity, accuracy, and all, right? And then you can tell that this method is good or not. So then we got servers to discriminate this type of the proteins based on the composition or the peptides and so on. Okay, it takes the sequence, right? Here it's sequence. When you predict here, this is your occurrence, this is your composition, this is your globular protein difference, this is YMP difference, this is less. So we can say that this is seems to be an outer membrane protein, right? You can calculate, you can predict, right? Fine. This is an exercise, right? So I give you two sequences. This is a sequence 1, this is sequence 2, this is the average composition for the alpha, this is for the beta. Take the first one, right? Can you discriminate, identify this belongs to alpha or beta? What to do with that? My first question is, I give the sequence, first sequence 3A0B, and I ask you whether it belongs to group A or group B, alpha or beta. What will you do? What will you do? To identify whether it belongs to alpha or beta? How will you group this protein, whether it belongs to group A or group B? What are the steps you have to do? Any comments? Any answers? Okay, so first you get the composition and compare with this alpha composition and compare with the beta composition here. Here alpha is 45 and beta is 70. So compare with these two, this is lowest, so this belongs to alpha. Right? The second one, the same, this is alpha is 36, beta is 18, so the 18 is less, so it is beta. Then we can also do the pair preference. So if you see the three pairs, right? If you see how far this AC or CA, AD, AE, and so on, right? So these are also you can calculate the residue pair composition, right? You can tell this also can be useful for discrimination. Okay. So here if you see how many times AA occurs, this is 4 times. 1, 2, 3, 4, and this is the number of times A plus number of times A. That is the 2A, so that's called 11.76. Right? So now accordingly you can see how different types of residue pair preferences. How many times this A preferred with A and D and C and so on. Right? This you can calculate. Then the next one is the profiles. You can calculate some profiles based on this sequence. Right? So if you see, this is the sequence, okay, SAMBLE, okay. So we have these numbers are here, right? So here the profile gives you the x-axis the sequence, and the y-axis the hydrophobicity values, right? So if you draw a graph connecting the sequence and the hydrophobicity indices, right? So first one goes with the -1, second one is 1, third one is 1, fourth is -1 and 2. So you'll get a pattern, right? I'm sure you are not interested to draw the graph now, right? So I don't want to ask you that. You can draw this and you will find a pattern. So that patterns are very important. The patterns will tell you that whether these particular regions are from helix, or particular regions are beta strand, or particular regions have any transformer segments and so on. Because these patterns are very important. That gives you a good information, right? So this is the numbers I gave you because just to draw a graph, but you are not interested, I'm sure. So I don't want you to ask you, right? So actually this is the actual number we have to plot, right? So this is the experimental value we obtained from the thermodynamic experiments, and this is the number we obtained using the computational analysis. So we can use these numbers to make a graph that will tell you the specific patterns. Okay, so now you can have the you can draw the graph. So right in the online. So use these values, which value you want. So if you take these values, right? Okay, the question is whether you need the plot with smoothing or without smoothing, right? So without smoothing, if you do, right, this is the graph, right? So you get the graph, the x-axis is the recipe number, y-axis is the hydrophobicity value. If you plot against each one, right? So now you will get this graph. Fine. So now you can smooth it, right? So you can smooth with any window length, for example, 2 or 3 or 5 or 7 or 9, whatever numbers you can give it here, right? So now you can give a smooth graph and you can see some patterns here. Okay, here this one stretch is completely above this average value, and here is something above the average value here and here like this. Okay. So these segments which are above the average value, right? So they are the transformer segments which cross the membrane. Right? Actually, there's 5 segments. This is what we're getting here: 1, 2, 3, 4, 5. Right? So we can get it, right? So this smoothing this graph, we easily predict. You don't have the structures, just we have the sequence. You can make a plot, and using the plot, you can easily predict where are these transmembrane segments and which are the segments which cross the membrane in the case of these transmembrane helical proteins. Okay, so another parameter that is called ambiphysicity, right? So that will give you the periodicities of the polar and nonpolar character, because some of them are polar, some of them are nonpolar, whether there's any periodic arrangement, right? Okay, so now this is the periodic arrangement for the helices. That if you see, as I told you, if you draw the figure, if anybody drew the figure, I can help you to get these segments there. Okay, so if you have this type of patterns, right, then you can see that this have the potential to have an alpha helices, right? So 2 in the one side, 2 in the other side. Why 2 in one side, 2 in the other side? Because in alpha helices, there are 3.6 residues per turn. And if you take this 2 phases, one phase contains 2 residues, which is highly hydrophobic, and the other phase which contains 2 residues, which are highly hydrophobic. That is the reason if you take the average here, 2 are hydrophobic and 2 are hydrophilic. So you can get your pattern, right? These type of patterns, then you can see that these are the transmembrane helical segments. So like this, you can get a beta strand, right? Here this alternating hydrophobicity. Right, you can see one in high hydrophobicity, one in low hydrophobicity, right? So here, if you draw a graph, and if you have this type of patterns, then you can tell that okay, this is the protein segment which has the beta strand, right? In this segment and in the beta strand, right? Oh, we have some equations here to calculate, but anyway, okay. How this is very strong or it's very weak. Based on these numbers, you can tell you the strength of this ambiguity character. So you can have some patterns, right? You can have some patterns here. So if you have this sequence, right? X-axis is the sequence and y-axis is the hydrophobicity, and I have the numbers for all these residues. And if you make it, so you will get this type of patterns. If you take this one, right, you will get this type of pattern. Right? If you substitute the values, right, so you get this type of patterns. This we can tell you this is mainly the transmembrane helical segment. This you can see this alternating hydrophobicity, and this belongs to the beta strand. Okay, I think I'll stop here and give you a 10 minutes break, and we'll start at 3:30. Okay, I think we shall continue. I'll are you able to see my screen?
Yes, sir. Visible.
Okay. Okay, good. Yeah, fine. So I show a couple of graphs, right, with respect to these sequences, right? So each one we have the numbers, we have the values here, and if we substitute the values, you'll get a pattern like this. Right, left side, if you see, is totally hydrophobic, so this mainly they passing the transmembrane regions. And the right side, if you see the patterns, right, this is only high hydrophobicity, one is low hydrophobicity, and you can see this periodicity here. And this is the ambiguity characteristics of beta strands. And if you see these type of patterns in your protein sequence, you can say that region could be a potential beta strand. Okay, now this is the definition for pattern, right? If you see here, we use the square brackets and we use X, and we sometimes we use some residue here, just G here, and sometimes we use the curly bracket like this. Okay, the square bracket means the residues which are inside this square bracket is allowed. For example, if I see LIVM, one of the residues in this 4 is allowed. And the second one VIC, one among the 3 is allowed, right? So what is allowed residues in the second one within the square bracket? Right, one among the 3. What are the 3 residues inside the square bracket? VIC, right? So one of the 3 is allowed. So then 2 gaps, 2 gap means you can put any amino acid here. Right, with among the 20 residues, you can have anything. And the next one is G, that is highly conserved. Right, so we have to keep C like that. G like this. Then here, one among these 3, these 6, and X is any residues, like and so on. If there's a curly bracket like this, that means that within that bracket, what are the residues, they are not allowed. Other than that, anything is fine. Right? So now take the first example. Okay, here, can you write a pattern using this expression? What will come in the first position? Can you write one residue for the first position? There is LIVM, one among these 4. So then what are the options? Right, we can use one of these 4. Either you can add L or I or V or M, any one of these 4. And what is the residue which occupies position number 2? B, correct, fine. Then the third position and fourth position. Okay, fine. And the fifth position? G, right? Fine. So now you can make several sequences using these specific patterns, right? These are definitional patterns. Okay, so now this is the pattern. Okay, so 17 amino acid prepared because it has 17 positions here. LAVRM position number 1, VIC position number 2, any residue position 3 and 4, G8 position 5, and so on. Right. So now you can search these patterns. So we have the PIR. So go to the search, and the pattern search is here. Just to click here the pattern search, right? Then this will give you, we ask for the pattern. We give the pattern. Here, this is the pattern we gave here, and click here in the submit button. So you can see there are various options. Okay, these are the sequences which contain the same pattern. If you click one of these links, right, we get the sequence here, and if you see, this follows this particular pattern. Right, LISQ, can you see verify this follows this pattern? First one is L, yes, within this. Second is I, it satisfies. H and Q, G, yes. E is here, K is here. Right? Then A is here, 2x. And here 4 times any one of these 6 residues. Right, K, L, Y, Y, I. Right? And then 2x, right? And G. Right? So it follows the pattern, right? This one. Right? Okay. So now we identified a specific motif. There is this pattern, right, for the beta-barrel membrane proteins to insert the peptides inside the membrane. Right, there were specific patterns. So if you see, this is one among this one, and then G, because G is small residues and highly flexible, and sometimes it is highly conserved to particular positions. And one among this here, and here dot means any residue X here. Here also dot is here, it's X. Right, here is one dot, and now this is this, right? So you can write a program to get this motif and why this is important. So I show the results. Okay, so here you can see these specific patterns, right? The pattern, if you see, it is only for M.B.A, M.B.A, ID90, right? If you see here. So this is the out of 73, 68, and the whole sequence is 73 and 73. So if others, if you see, only 2 out of 66, 4 out of 34, 32 or 293, and so on. There's everything is less than 0.2 or 0.3, and this signal, if you see it is 0.9, that means that beta signal is very specific for the beta-barrel membrane proteins. That we could publish in a paper called Cell. There's impact factor more than 30. Fine. These are new signal we proposed after that. So we included some other amino acids here, and we included the new signal. That also get better results than the beta signal. Okay, now the question is, okay, we had done all these things, but now when you do the analysis, when you go to real analysis, right, you get the data from different databases. What is the database I discussed for protein sequences? What's the database for protein sequence I discussed earlier? Do you remember the database for protein sequences? Right, UniProt, right? PDB is for the protein structures, and the UniProt is for the protein sequences. So if you use all the sequences in the UniProt, there are various sequences which are highly redundant. So if you use the redundant sequences, it will be overrepresented. For example, if we have these 2 sequences, right? ADIK, this KL 2 sequences. If you take the average of A, totally 10 plus 10, 20. Out of 20, how many A occurs? 4 times. So 4 by 20, that is equal to 0.2. If one of the sequences appear twice, for example, this I put 2 times here. In this case, average A, if you see, there is 0.23. And if second one occurs 2 times, then the average A is 0.17. So if you put the same sequences again and again, that will introduce a bias, that will be overrepresented. So then in this case, you get a result, it's biased with these overrepresented sequences. And if you develop any algorithm, that will work for the training set, but not for the test set. Right? So in this case, you need to reduce redundancy. So there are various methods to reduce redundancy, and these are the three commonly used programs, right? The CD hit, blast class, and PISES, right? It is the based on this the sequence matching, right? So CD hit is very popular because there is a cluster database with high identity tolerance. It takes a faster format sequences and the non-redundant sequence as the output. It's very simple. It can handle a huge data set. It is easy to download, and you can get the results very quickly. Okay, so this is the grid increment algorithm, right? So in this case, the sequence with the identity of more than specific threshold that will be discarded. They take the largest sequences first and proceed with the shorter ones. How to get the sequence identity? They take the number of identical residues, right, and divide by the length of the shorter sequence. Okay, so now we have the different algorithms. Because the full sequence is time consuming, so they divide into pieces, right? So take the 10 residues, right? If it is more than 90% sequence identity, you find at least one match. Because it is very rare to have the 10 residues with the same sequence. If you find that, then they assume that the sequence have more than 90% sequence identity. Like the pentapeptides, it will be present, and they consider more number of segments for the particular pentapeptides or tetrapeptides and so on, right? So they use the clustering methods, right? So they use a k-means clustering, right, to have the n observations into k clusters. So first we put it here, right, and then we move it here and there, and finally they made into the different clusters and take the random sequences from each one. That will give you the non-random sequences because they are completely different clusters. So if anything between this cluster 1 and cluster 2, the difference is more than the threshold, right? So in this case, you can take the non-random sequences. So there are various ways to get the clusters. So we can use some properties. They can use the Hamming distance of the composition, including distance, right? So various ways to do it. This is to how to get the Hamming distance. You can calculate for these three and see which one is close to each other, right? Anyway, I don't ask you to do it because you won't do it. So I'll pass through this one, right? So then the Euclidean distance, you can use this equation and then see which one are close to each other. And in this case, you can make two clusters, one for one side, one for the other side, and among these three, right? If you use this sequence. Fine. This is the installation for the CD hit. You can get it from the website here, and then you can use this command to run the CD hit, right? This, for example, so if you run it, these are your sequences, and finally you get the sequence, right, for when you run this CD hit. So PISES is another software which it has pre-assigned sequences. Either you can use the existing sequences in that PISES server, or you can make your own sequence, right, that accepts everything. Okay, so here is the website, and you access the server to create your own list, right? So here you can give your sequence in the FASTA format, and then here this is your sequence here. Ask for the maximum percent identity and the chain length, right? If you click it, and you get the sequences, right? This is the sequence with the sequences, and here you give the IDs. So these are the IDs for your sequences, right? Fine. So now the next question is, okay, if we have two sequences, right? How far they are similar? Can you tell which regions, which residues are similar between these two sequences? Some regions are similar, right? If you see the sequence, and you can see that. Right? Yeah. So you can see some sequences, right? They are similar. But easily, if you see what is the percentage of similarity or percentage identity, that you cannot say like this directly, right? Fine. This alignment sequence tells you how they are related to each other, and what is the evolutionary history of these sequences, and how they evolved to get one sequence to other sequences depending on some specific period of time, so that you can understand the information content as well as the function of these genetic sequences. That is the advantages of having this alignment between these different sequences. Fine. So there are various ways to do this alignment. The simplest one is a dot plot that will tell you the basic information why are these two the sequences, they are similar to each other. So they have local alignment, so you can see the sequences with the partial homology, and you can global alignment, compare the full sequences and see what is the identity between the two sequences. So this is our plot, right? So you can see the x-axis, you can see this one sequence of the x-axis and one sequence of the y-axis. And if you see the residues are the same, then you put a dot. So if this is the x-axis and this y-axis, V and V match, so put a dot, and this V will match with another V here, another V here, then you put a dot. So then the second position, you put the dot, right? So now we can make a dot and you see where we can see the continuous set of lines. The continuous set of lines, then you can see that, okay, here this segment are same, right, in the case of the sequence 1 and sequence 2. They're same. Then similar one we can do, we can adjust to the similarities, right, isolation equivalent to valine, because both aliphatic, leucine and isolation, and the origin and lysine. So we can do it, and then we can give the adjustment so that you can see the similarity. You see here, there's a diagonal, you can see this stretch of residues, right? You can see there you have the lines. That means these residues are similar, the same in these two sequences. If you see here, this is a region here. Okay, here this region. So that is the same. Here you can see the alignment. If it is above the diagonal, that means that there is an insertion in one sequence, and below the diagonal, that's the insertion or deletion in another sequence. That will tell you whether it's insertion or deletion in one of the sequences. Okay, so now we have two sequences. One is the human, one is the chicken. So these are the two sequences here: the human hemoglobin and this is the chicken hemoglobin. So how far they are similar to each other? So we can use this BLAST. That's a program, right, to compare these sequences, right? If we give the sequence 1 here and the sequence 2 here, and okay, you can see this is alignment, and this is your identity, and this is your positives. These are the gaps, right? What is the meaning of identity here? What's the meaning of identity? The exact match. Okay, so for example, if you see M and M, you can put the same residue M here. So with the exact match, that is identity. That means out of 147, 1 or 2 exact match, that is 69%. Positives means that is exact match plus similar. For example, isolation valine and the lysine arginine, aspartic acid, glutamic acid. That's you can see the plus here. Right? There's a plus means okay, these residues are similar in some characteristics. They're polar, or they can be substitutable using the PAM matrix. Right? It's possible to substitute. They say to plus. If you count the plus and the residues, there are 121 out of 147. That is 82%. Gaps means the alignment where is any gap. There's no gap, so it is 0. And the query coverage, you can see either if some residues are missing in the N terminal or C terminal, that we remove, and others we consider as a query coverage. In the multiple sequence alignment, that is a pairwise alignment I discussed now. Then we go with the multiple sequence alignment. Here they have various sequences, many sequences. So that you can try to align. And here if you see position number 9, all are A, so put as A. Here some of them are B, right, and some of them are L, so either B or L, right. So there is you put the small letters, the capital letters are the same one, you put the capital letters here. So when you have the multiple sequence alignment, you can check whether any residues are conserved. Conserved means the same residue occupies at the same position in different sequences. If you see 5 sequences, right? So what are the conserved residues here? Can you tell the conserved positions and the sequences? What are the conserved residues? Right, G3, G4, and V7, and this A9. Okay, these are conserved residues. So how to calculate the conservation? So why do you concern about conservation? This conservation means if it is highly conserved, that residues are important. And if you mutate these residues, that will severely affect your structure and function. So if the conservation score is very high, then that residues are not good to mutate, because that will alter your structure as well as the function. And if you compare different sequences, some residues which can change quite often, that is called variable, right? And some of them are completely, they is not changed at all, they maintain the position, that's called conserved. This is mainly depends upon the evolutionary rate. How far there is a change of amino acid from one sequence to another sequence, right? This can compare the homologous sequences in the person PDB or the UniProt, and then see what are the residues which try to change, which residues tend to maintain at a particular position. Okay, so I'll tell you some algorithms you can develop, right? So you can get the data you can derive from these sequences, right? I have some 10 sequences here, right? The multiple sequence alignment, right? You can calculate the consensus score based on various aspects. There are two major steps. One is get the frequency, position specific amino acid frequency, and the second one is the configuration score. There are various measures. Here this is a weighted frequency, unweighted frequency, or estimated independent counts. And the configuration score, you can say entropy based or variance based or sum of based method and so on. And this is what we can do that. Okay, you can use unweighted frequency. So that's given as FA of phi is given as NA of phi divided by N of phi, where NA of phi is number of sequences, so which post I is occupied with amino acid A, and NI is the total number of aligned sequences. So this you can calculate for the frequency of this any particular position. Then the entropy based measure, right? You can calculate convert the frequency into score. Right? This is given as f of a of phi. There is a frequency into a logarithmic of this frequency, that is the entropy based measure, where this varies from 1 to 20 or 20 amino acid residues. Okay, now I have the data. For example, take question number 3. Right? Question number 3. Which residues are occurred in question number 3? And how many times each residue occurs in question number 3? Division, how many times? Ten, right? Take question number 6. What are the residues present in question number 6? And how many times the residues occur in position number A? How many times? Because I need the data for the calculation. Okay, A8, then what else? What are other residues in that position? And how many times? No, I am talking about the position number 6. K 1 time, not 10 times. K is this position number 6. If you see here, B 8 times, position number 6, 3, 4, 5, 6, A 8 times, then E 1 time, and D 1 times. Right? So 8A, 1D, and 1E. Okay, now you can calculate, right? So you see here, question number 3. What is the frequency of question number L? That is 10 by 10, that is equal to 1. Right? We take question number 6. E occurs 8 times, the frequency is 0.8. D occurs 1 time, frequency 0.1. E occurs 1 time, frequency 0.1. Now we convert this frequency into score. The formula is, this is a formula, FA of I into logarithmic of FA of I, right? So here take the question number 3. FA, what is FA of I? That is 1, and logarithmic of 1. Logarithmic of 1 equal to 0, so finally this number is 0. Can you calculate question number 6? What is the conservation score for question number 6? Right, you can do that. 0.8 into logarithm of 0.8, 0.1 logarithm of 0.1, 0.1 logarithm of 0.1. If you calculate all these things, you get minus 0.638. Okay, so this is software where you can calculate, give a sequence, right? And then you submit here, right? So we get this position number 3, we got 0, squirrels we got 0. Position number 6, 0.638, so we got 0.639, right? So you can get the number. Fine. Okay, so now you can also normalize the value using the x minus x bar by sigma, and then you can get the normalized value. Okay, this is regarding the protein sequence. So we can do various aspects of the protein sequence, right? Now we can also have a lot of information structures. What are the different levels of protein structure I discussed? What are different levels of protein structures? Primary, secondary, tertiary, and quaternary. Primary structure we sequence that we discussed a lot. Then now I will show you some information regarding protein 3D structures. Okay, tertiary structure provide information right regarding the atomic level details, right? So you can give the PDB. If you see there's a Cartesian coordinate system, they give the XYZ coordinates. Okay, there are various experiments like X-ray crystallography, NMR spectroscopy, electron microscopy. These are the various techniques which are used to determine the 3D structures. And X-ray crystallography, you see they involve various steps like protein purification, crystallization, crystal mounting, data collection, and data processing, and the structure refinement, and finally you can use test analysis. So you get the crystal here, this electron density map. They convert the electron density map to the 3D structures. So these structures are deposited in the protein data bank, right? So PDB. The PDB earlier in 1977 they started to collect the Brookhaven National Laboratories with this protein structures, and then they formed the research collaboratory for structural bioinformatics, the consortium, and they developed protein data bank, and they have mirror sites in PDB US in the San Diego Supercomputing Center, and then PDB Europe, EMBL, and the PDB Japan, the Osaka University. So they are together, they meet together, that the deposition, processing, and distribution. Why we are so much interested in structures? Why we are so much interested in 3D structures? Because structures are important to get a function, right? And you can see there are so many number of prizes. So you just to solve the structures, starting from this 1946, this is discovery of enzymes to be crystallized, right? And then you can see the Watson Crick for this nucleic DNA structure, and Dorothy Hodgkins for this vitamin B12, and Christian Hankins and Aaron Clark for the protein RNA complexes, and Dyson-Hofer's group for the membrane proteins, and you can see Kurt Wittrich for the NMR, and you can see Venki Ramakrishnan group for the ribozymes, and 2012 GPCRs, and 2013 the MD simulations, and 2017 the cryo-electron. So there are so many Nobel Prizes solving the structures of proteins to understand the functions. So here, if you see, there is currently 170,000 structures here, and this is based on mainly on proteins and some protein nucleic acids and some other complexes. And most of the structures are obtained from X-ray crystallography, followed with NMR and electron microscopy. So in the COVID-19, there are various structures in the COVID-19 structures in the protein data bank, right? So we see this is 6LE7, the main protease with the inhibitor. And here this is structural similarity with the SARS-CoV-2 and SARS main protease, right? So you can see they are very similar. And this is your dimer interface. You can see this is the region where they interact with each other. Right, so we also did some analysis. There are various analysis going on SARS-CoV-2 to development of the vaccines and the drugs and so on. So we did drug repurposing, so to check the different already available drugs, lopinavir, oseltamivir, and ritonavir, and we could see that the synergy of these drugs, not only one drug. This is mainly used for the HIV, and one is for the flu. So we can see that the combination of the drugs works better. We get a better binding affinity of -8.3 kilocalorie per mole. Like this, we are also working on various aspects using the protein structures to identify the mutations, which mutations are virulent and which are not virulent, and so on. So now you can see the protein data bank. Right here, you can all the structures of the proteins, and you can see the XYZ coordinates. So these are the location of each amino acid. These atoms, here is the heteroatom, that is mainly the water molecules or the ions here. And they give one column called occupancy. Occupancy is 1, if there is only 1. With they occupy 2 positions because of these ligands, they have the interaction ligand, they occupy 2 positions. Total will be 1, but they have 2 alternate positions. So here if you see 0.5, 0.5 or 0.4 and 0.6, right? So that sum is equal to 1. So here if you see 0.43 and 0.57, so the total will be 1. They have 2 different conformations. The temperature factor will tell you how far the fluctuations of each residue. So whether they are able to fit within the electron density map, that's very rigid. Here if you see the blue ones are the low values which are rigid, and the yellow ones which are very flexible. If you are not able to fit in the electron density map, they're completely flexible. So you can see the orange one and the red and yellow one which are highly flexible. So now you can visualize these proteins. There are various software available to visualize these proteins. Say Pymol, Rasmol, Jmol, and so on. This is very good software, and Pymol is one of the best software, right, to view the structures, and you can calculate various parameters using 3D structures. I'll show you one demo using the Pymol. Okay, this is your installation, how to install that. And here you see 2 windows. One window is here. This is the you can write any commands here, and you can see this is the graphic interface. So you can do that. And this is a command prompt, and you can see various menu options here. And here is a viewer window. And you select any PDB to view. So go here, open, and you open the file. Okay, it's here, and go to the file. So you get it. Right. This is default view. You get the lines. Right. So there are different views you can get. Right. So if you see here, right, you see here ASHLC. Right. Here the A stands for action, S stands for show, H for hide, L for label, and C for color. So this is the meaning of this. Right. So here we have the lines. Right. You can see as cartoon. Maybe you can show as cartoon is here. So label is cartoon. So if you take the cartoon, so you will get this cartoon type structure. Okay, then you can color by chains. Here you have how many chains in this protein. Are you able to identify how many chains in this protein? 1, 2, 3, 4. Can you see the structure figure and then see how many chains? Right, so you're not able to tell anything. Okay, so if you do it, so now you can see the colors. So two chains, one chain is here, one chain is here, right? One team, there is 2 chains, right? Now also you can remove one chain, but hide chain B. So right click on the chain, right, and then you can hide everything and you can get it, right? So here you can see, right, this is the chain, right? So you can get the helix, sheet, and loop. You can see the different secondary structures, right? I discussed earlier, right? So you can different colors. You can see this helix as red, and the strand as in yellow, and the loop as green. So you can change the colors if you want. So you can do it. Right? You can see this is sheet, and here this is the loop. Okay, now you can give the sequence for the protein sequence. You can give. You can now see it's shown here. So you can get the sequence. Right? You can get it here. The colors match. The red ones are here, the helix, and the yellow ones are here, the strand, and the green ones are here for the loop region. Right? Fine. So you can see the color code, and this helix is here, strand, loop, and everything is here. Right? So now you can display the color scheme with chain. Right? We see some group. You can take this, right, for better information. Right? It's here. It's marked here. So now you can calculate and you can analyze much more details about any particular regions if you want. You can zoom it. I can hide everything, and finally change the sticks. So you can see the ball and stick model. Right? You can see the carbon is in green, nitrogen N and the oxygen O. That is the notation generally used. So you can get it. Right, so you can label the residues. So this is 23 and lesion 24 here, and you can label the residues, right? And we select a lesion 24, and you can receive the atom name. You can label. This is CA, C alpha, C beta, N, C O, C gamma, CD1, and CD2. So now we can see the backbone atoms. Okay, here, and these are side chain atoms. So we can classify in regions, right? And we can show it a line. So this is now it is line. Then you can also measure. You can measure the bond distance, bond angle, and diagonal angles, right? So if we have the bond distance, right? How many atoms are required to get the bond length? 2, right? So if you click 2, right? If this is the first atom, the second atom, so this is 1.5 angstrom, right? So how many atoms are required for the bond angle? 3 atoms. So we can check here. This is the first atom, the second or the third atom. So this is a bond angle, that is 109.9 degrees. So right, you can write the V factors, the temperature factors. You can see that, and you can show this surface diagram where you can see the different colors, right? So blue is the least flexible, and orange is highly flexible, right? Then if you want to know more about the PyMol, so you can compare the different structures. You can do the mutations. You can get the high resolution structures, and you can support Python scripting for all user needs, right? You can get the interface residues, pairs, distance, and so on. Okay, if you want to know more about the PyMol, there are tutorials available. Just you Google it PyMol, and you can get the tutorials, and you can get it. Fine. So now if you have a structure, what can you do with the structure? With the sequence, I explained various aspects that what can we do with the sequence. Now we got 3D coordinates, right? So what can we do with the structures, right? We can do so many things. So first one is we can see which residues are in contact, right? Whether residue 1 and 10 are in contact, 1 and 2 are in contact. So if you for this contact, we need few information. One is the distance. What is the cutoff distance you want to take? 5 angstrom or 6 angstrom or 7 angstrom or 8 angstrom, right? If you see, then second one is which atoms you want to consider, whether C alpha or C beta or any heavy atoms, right? You see, x-axis is the residue number, y-axis is the residue number, 1 and 2 are in contact to put it out, right? So say 8 angstrom and with the any C alpha atoms, right? So then you can put a dot if it satisfies the criteria, otherwise just leave a blank. So now if you see the figure, right? Can you see this figure and what can you tell from this figure? Can you tell something from this figure? Regarding the contacts, where are the contacts? Mainly the contacts are located in which place? Can you see the dots? Are you able to see my screen? Where the dots are located? Where you have more number of dots, where you get less number of dots? Yeah, if you see the diagonal, you can see they are highly dense. Because there is a residues which are close to each other. Because 1 and 2, 2 and 3, 3 and 4, right? They are always in contact. So that is the reason why you have the close contacts in the diagonal. And there are some residues which are very close to the diagonal. There is 2 and 5, 2 and 6, like that. The residues which form alpha helices, right? Right, so they are formed by these hydrogen bonds. So they are very close to the diagonal because they form I and I plus for hydrogen bonds, right? And they are very far away in these some cases. You can see the 10 and 400, 350 or something. So here they are very far away. But in this case, they are mainly the beta strands. Okay, in this case, they are very far away in the sequence, but they are close in space. So when you make a structure, this contact map, this is a kind of 3D structures into 2D form. Right? Easily you can tell that which residues are in contact in 3D structures without looking into 3D structure. That is called, we have the contact maps. Right? So we have the, for example, I have the 10 atom, 10 residues, 1 to 10. These are the coordinates that I ask you to construct a contact map. Right? I'm sure nobody will be interested to do the contact map, so that you take it as exercise. So if you want, you can do it at home. Okay. So now if you do it, you can see some of them are very close in space. For example, here these are residues which are very close in space, right? And some of them are very close in sequence, and some of them are far away in the sequence. Right? These are two concepts. One is residues which are in close contact, and the second one, they respect the location of the sequence. So based on that, so we classify into 3 different contacts: short range contacts, medium range contacts, and long range contacts. Short range contacts is between less than plus or minus 3 residues. Medium range contacts 3 or 4 residues, and long range contacts plus or minus 4 residues. And we can also further divide in small bins of 10 residues. If you see that 352 are the central residue, can you tell one of the short range contacts this T152 can make? Can you tell one short range contact with the T152? T150. Okay, fine. So can I tell one medium range contact? This is given in the figure itself. Well, 149 because it has 3 or 4 residues, right? One long range contact. A98, right? So it's very far. So if you see, they are very close in space, but they are about 50 to 54 residues far apart, right? So they are sequence-wise, it is very far, but they are close in space. So these are long range contacts, right? So this we can show in the contact maps. So here this is the short range contact. This is the on the diagonal, and the next to diagonal, and the other one is close to diagonal. There is a medium range contact close to diagonals, and the far away from the diagonal, they are the long range contacts, right? So stop. Now the next concept is accessible surface area. That will tell you how far these residues in your protein is accessible to solvent. If this is the protein chain, protein structure, okay, I put the interlocking spheres, and we take one water molecule of 1.4 angstrom and roll the water molecule on the surface of the protein and see how far this can penetrate into the van der Waals surface of these residues or each atoms. Right? If this outside, which one has high surface area and which one has low surface area? Because if we have two residues, one is at the surface, right? One is at the buried interior. Which one has high accessible surface area? The one the interior or which is the located surface. Right, in protein there are several residues. Some of them are buried interior, some of them are located at the surface. Which one has high accessible surface area? The interior ones or the outside ones? Outside ones, right? Because I give you some numbers. Okay, this I skip. Okay, so if you see here, can you tell which these are the residues here? Which residues are interior? Which residues are at the surface? Can you tell one example? Residues which are located at the surface and which residues which are inside the protein? The numbers are here. Accept surface area values are here. This is the amino acid residue. So which residues are inside? Which residues are outside? E and D are surface because you can see it is 146 and 1414. Yes, and the inside ones. You can see this is A is 0 and this is 1 and this is 0. So these are the individuals, right? So here you can easily get the numbers. For the further large scale analysis, you can use that. For the biology, it's difficult to get the numbers, to deal with the numbers. So it is easy for them if you show in time of pictures. So here I show the pictures. Okay, take one protein. So I got the values and we showed in pictures. Right? If you see, can you see the colors? Different colors. Yeah, different colors represent different types of amino acids. For example, the blue is the positive charge, and the red is negative charge, and the gray is the hydrophobic and so on. And the size also different. The size depends on the area, actual area. Bigger size is higher surface area, and smaller one is a small surface area. You see, most of the gray are inside the protein. The hydrophobic residues located inside, and some of the polar residues and the charged residues outside. They have high surface area, right? This thing. So you can do it in the amino acid sequence wise. You can see the lower ions here and there, but they come close to each other. They form the hydrophobic core. Okay, so here we have the program. Just you can give the PDB ID and get plot. They ask which plot you want, right? See the spiral plot, or you get the sequence plot, so you can get that, right? Then we have the link to the actual numbers, and you get it, right? Okay, now we can get the percentage accessibility. That means we tell you, okay, with respect to the unfolded state, that means it is completely elongated chain. How far this one is the exposed or buried or partially buried, right? Fine. So the next one, you can calculate the hydrophobicity. So this is the hydrophobic behavior of amino acid residues in a protein, right? So we get the hydrophobic values based on the thermodynamic transfer experiments like the ethanol water experiments or octanol water experiments and so on, right? So then also we reported that the hydrophobic behavior of this residue, it depends on the residues which are surrounded by that particular residue, right? That means the hydrophobic behavior of this T is mainly depending on the residues which are around this limit of 8 angstrom from that particular residue, right? So if you see that, so we have the experimental values. These are not experimental values. This is I put for reference. And I showed earlier about the two scales. One is my scale and the other one is the transport scale, right? So now to get the T, so you see the residues which are occurring in this limit and get the values from here, right? And can you tell what is the hydrophobic value of the central residue T? Can you calculate and get the number? What are the residues occurring within this 8 angstrom limit of this T? These are the residues T, R, D, A, F, B, L, right? So you get the numbers from here, and can you tell the value for this T? Sorry. Two, yeah, but the D is -2, R is -2, so -4, -5, -4, -2, 0, 2, right? 2. If you can take this T, this is also same as 2, or this will be different. Yeah, that is 2. Correct, good. So if you take this T, then the value will be the same 2 or will be different. That will be different, right? Because to say for this one, you have to construct another sphere of 8 angstrom around with this as a central one, and you check the residues which are occurring within that limit, then get the numbers, and finally you get the different numbers. That means the hydrophobic behavior of this residue depends on the surrounding residues in this particular environment, right? There is another concept, there is a contact order. Contact order tells you right? How about the local and non-local contacts, right, in a protein structure? For example, if you see this T1 and F7, the distance of 6, right? And for each one, we have the different distances, right? So it's calculated using this formula. CO is given as sigma delta Sij by L into N. Sij is the distance of separation, sequence separation. Okay, T1 and D2 is 1, T1 and R4 is 3, L3 is 2, T1 and F7 6. Add up all the numbers. Divided by L is total number of residues, and N is total number of contacts. So if you do this, then you will get a total contact order for a particular protein. If we more number of long range contacts, contact order will be less or more. There's more because this number is different. If it is more, then automatically this contact order is more. Right? If it is less number of long range contacts, then the contact order is less. Then there is another concept called long range order. Long range order will tells you the contact between the residues which are close in space and which are far away in the sequence. Right? This is given as Nij by N, where Nij equal to 1 if the modulus of I minus j there is more than 12. How we got this 12? Okay, that I show next slide. Or it is 0 otherwise. So if you see this, so what is value of Nij in this case for this T1? How many residues for the distance separation of more than 12 residues? Two, right? Because see, these are the two, 28, 50 and 28, right? Fine. So now do it for all the residues, go 2, 3, everything, take the summation, and N is the total number of residues in a particular protein. So how we got this number? We gave the 23 two-stage proteins. We know the value of these folding rates. So we change these numbers, contacts, and we get the correlation coefficient, and we got this best correlation at number 12. This is where we use the cutoff of 12 to define this long range order. So we did for the different structural classes, alpha, beta, and mixed class, and we get the good correlation. This is the relationship contact order long range order, and you could see here you can do it the good correlation for the mixture, but here it is less correlation. But here we could get the good correlation for all these classes. Okay, now skip this. So the electron, they use these parameters, right, to do the analysis, and we found that this long range order you could get good correlation in almost all the classes, right, compared with the other parameters like chain length or relative contact order, absolute contact, and so on. Okay, this is second aspect. First, I did the contact maps with surrounding hydrophobicity. We did the contact order and long range order. Okay, the last one I talk about the binding sites, right? Okay, how to define the binding sites? Okay, so if you see the complexes, protein interact with the proteins, protein interact with the nucleic acid, protein interact with the carbohydrates, protein interact with ligands. Take a protein complexes, how to identify the binding sites. Right? If you see the complex is here, one is in green color and the blue color, and we cut it in two pieces. Okay, one is here, one is here. And from any atom, any heavy atom here, any heavy atom here, are C alpha atoms. And see any cutoff. You can see any heavy atoms of 5 angstrom, C alpha atoms of 6 angstrom, C beta atoms of 6 angstrom, right? So if there is within this distance, you can use this equation to calculate the distance. And if any two atoms are within this limit of 6 angstrom, then we can see that okay, these two residues are at the interface. Here I show an example. This is the PDB ID, PDB code, PDB coordinates. This is chain A, this is chain B. Here arginine is here is aspartic acid. Right here, if you see these two are within this limit of this 5 angstrom. If you calculate this one, right? So if you put the details, okay, these forms the interleukin receptor complex and forms electrostatic interactions between arginine 88 and aspartic acid 72. This is the explicit coordinate. What is the distance? This is explicit coordinates. This is explicit coordinates. What is the distance between these two? That is less than 5 angstrom, right? So in this case, you can say that these two residues, they are having the interactions, right? Because the distance is less than 5 angstrom. Okay, we developed a server, right? So to identify these interacting residues. So here, if you can identify the binding sites, and if you ask about the protein and chain name is the optional, and get result of 3.5, right? And we'll get the result. Okay, these are results within 3.5, and here these are the one which are interacting to each other, right? Fine. So this is one method to identify the binding sites. The distance between the two residues in the two proteins, right, which are within this specific threshold of 6 angstrom or 5 angstrom, depending upon the atoms you use, then they are together. And the second aspect is accessibility that I discussed about the concept of accessibility earlier. So we have the chain A, chain B here. We cut into two pieces and get the accessible surface area of this chain and with the whole complex. Right? So if you see this region, so the accessible surface area are same or different here and here. We don't change any conformation, just we keep it. Just cut into two pieces, right? So here this outside, if we calculate the accessible surface area here and here, so we get the same or different. Any idea? You get the accessible area same or different? Yeah, because here is these are the exposed to solvent, here also exposed to solvent, you get the same numbers. So, but if you go to this area, right? So here this area is completely exposed. So here this area it is this is interacting with this protein. That is why here there is obstacle here. So in this region, if you take, you get the same area, different areas. So it's different, right? Because here nothing is here, so it's highly accessible. Here it is kindred with this another protein. So here the accessibility will be less because you cannot access to the solvent, right? So now if you compare these accessible surface areas, if there's a change in the accessibility, then you can say that okay, these residues which are the interface, because upon binding it reduces the accessible surface area. Right? You see here, this is the one of the complex. This is the one with the free protein. Free protein is very high. Okay, this complex, it is completely zero, completely buried. So here are the region where this is at the interface. And another approach, I'll stop now, or maybe another 1 or 2 minutes I'll finish it up. So we use the energy-based approach to calculate the interaction energy, right? And then you can see the residues which are having the less than 1 kilocalorie per mole, right? So calculate the energy, and if it is less than 1.1 kilocalorie per mole, so they are identified as the binding site residues, right? So then we can compare these different types of concepts to get the interface residues. The three concepts we discussed. One is based on the distance, one is based on the accepted surface area, and another one is based on the energy-based approach. I think I stop here. If you have any questions, just you ask me. I don't want to explain all these things. If you want to have more information, just you check my book, and there you can find almost all the information. Okay, so these are my books. Protein Bioinformatics I published in 2010 by Elsevier Academic Press, and 2020 Protein Interactions by World Scientific, just last year published. Okay, if you need more information, just you can refer my books. I will get all the information. Okay, thanks for your attention, and I'll stop here. And if you have questions, I'm glad to answer. Yeah, we have all the slides. If you want to have the more details, you can check my NPTEL slides have all the information.
Participants, feel free contact to professor, discuss with professor. Okay. So no more question, professor?
Yeah, if there are any questions, I'm glad to answer.
Okay, professor. Okay, professor.
Yeah, even I ask questions, they don't answer. They are not happy to answer, so I don't ask.
Okay, sir.
Yeah.
Okay, professor.
Yeah. If any questions is fine.
Professor, no more questions.
Okay, that is fine. Yeah, yeah. Okay, yeah.
Okay, sir. So no more questions. I formally thanks to what of very today's session is very important session, and also Professor is came from IIT Delhi. So nicely introduction and explain to sequence, bank, and related to the proteins of structures. Thank you very much, Professor.
Fine, okay, yeah, good. Yeah.
Yeah, Professor
Thank
is
you.
very busy schedule. I don't know. I know a lot of tight schedule in there. So given the time, valuable time, Professor is given the valuable time and come with given the lecture is very great successful on today lecture, Professor.
Thank you.
Thank you very much, Professor.
Thank you. Bye. Bye.
Thank you, Professor.